Thanks for the introduction, Bill. I think, we think a lot about translational science and how do we move the needle from preclinical to the clinical trials to actually implementation. So we're going to be talking here with and having a discussion with 4 kind of experts in the preclinical, moving things into patients, and we're going to talk about barriers and I think have a discussion. So we will open up for questions, but I'm going to do a little bit of introduction from each of the panelists, and we'll ask some questions to get things started, but we want this to be very interactive, so if you have questions, think about them, and we'll circulate with the mic to get some questions, and we'll take questions also that come in online through the chat. So without further ado, I think we have, I don't know how many of these mics, I guess everyone almost has one. We will pass this over to, I'll pass this over to Rachel Presti, and just want some introduction of where, your background and what kind of work you do.
Okay, so I've been here for a long time, so my background is very long, but what I do right now is I run the Infectious Disease Clinical Trials unit. I do have a PhD in immunology and was in basic science for a while, and I'm very interested in translational research and moving things into clinical trials and clinical research.
I'm Christina Stallings. I'm a professor in the molecular microbiology department. I am a PhD straight basic scientist that studies the molecular mechanisms of mycobacterial pathogenesis. But even though we're doing very basic biology in my laboratory using culture-based systems and animal models, we have a commitment to applying what our basic science findings are to the development of new treatments for mycobacteria. And part of that is because tuberculosis kills more people than any infection in the world, and non-tuberculosis mycobacteria are a growing problem. And so when Bill was presenting the gaps, I'm constantly trying to traverse the gap between that, basic science research to actually application of clinical trials, clinical science, and translation to medicines.
Good morning, everyone. I'm Kory Lavine. I'm a professor in the Division of Cardiology. I'm a physician scientist, mostly heart failure and transplant specialist. My research is focused on myocardial inflammation. And like Christina, I mostly live in GAP 1. We're focused on using what we can learn from patients, their genetics, their tissues, their other biospecimens to learn about their disease, to develop new opportunities to try to create therapies, and then also spend time in early therapeutic development and clinical trials, mostly proof of concept in humans.
Hi everyone, good morning. I am Fumihiko from the Department of Medicine. I am a professor of medicine in the Division of Endocrinology, and my research team has been studying a rare genetic disease called Wolfram syndrome, characterized by juvenile onset diabetes, optic nerve atrophy, and neurodegeneration. And I have been conducting both clinical and basic science research and try to, translate the findings in the lab to treat patients with this disease. Nice to meet you.
Great. So when we think about barriers and challenges, I guess, Fumi, we'll start with you. Kind of what are the upstream factors that you've had to kind of encounter to move into the preclinical and clinical trial space?
So one of the major challenges to move from the preclinical stage to the clinical trial stage is to find to write up the clinical trial protocol. And to write up the clinical trial protocol, we have 2 different components. The first component is to show that the potential treatment is really has potential benefits for patients with Wolfram by showing the preclinical data in animals and also cells. And so to show, and we need to include that portion into the package to the FDA. So investigation new drug application to the FDA. And so that's the first part. And in the clinical trial protocol part, we need to identify or specify the specific outcome measures to assess the efficacy of any new potential treatment. And so to identify the best outcome measures, we need to know the disease clinically very well. And so we have been, my team has been working with the Dr. Tamihashi's group in psychiatry, who ran the natural history study from until 2023, and we utilized the data obtained in the natural history study to write up their clinical trial protocol. So my point is, I think the first challenge we usually encounter is to write up the good protocol, clinical trial protocol, to accurately assess the efficacy of any new treatment. And to achieve that, we need both preclinical data and also natural history data.
Great. Anyone else want to comment on challenges that they've found and ways they've encountered and improved the process?
Yeah, so I mean, the gap is actually a very, very big gap, and in my experience, there are two major problems in that gap, and one is funding, because it's actually and process. It's actually very hard to get funding to cross that gap. So a lot of NIH funding prohibits clinical trials, and so it's very difficult to move something with a single stream of funding. You need separate funding. You also generally need a partner, because there are huge regulatory issues with moving something from the preclinical stage into the clinical stage. So yes, you have to write a protocol, but you also need to take any intervention. It needs to be well studied preclinically for safety, efficacy, all of that needs to be submitted.
How did you get through that gap? What were there resources that helped you?
So I'm going to let other people talk about that to some extent, because I am on the other side of the gap. So I've helped people with developing protocols and with actually running the trials, and to some extent with like moving with the FDA. There are groups. There are definitely the ICTS. Our clinical research unit, other clinical research units are very happy to help sort of develop the protocols. It's really moving, and a lot of people at that point will move to industry or to CROs to actually do toxicology and to get all of the preclinical data ready to submit to IND. But here.
Yeah, so I completely agree with what Rachel said, and I think that also addressing Chris's question, I think this is where partnerships become key, because the gap is big, and I don't think anyone can do it alone. And so I think there are resources like through CROs. Also, the NIH has some resources in terms of PK/PD, ADME, like things like that, formulations, which is helpful. But you do need funding if you're going to use a CRO, so you still need to find a way to have that funding. And I think for some things, there's more scientific gaps, which partnerships are key for, like chemistry. So if you're developing new chemical, or you've developed some chemical matter that works, like there will be optimization that has to occur. And so having also some scientific partnerships as well. And so I think it's a team effort to get there that requires experts in so many different areas. And so using resources at like ICTS, the NIH, is critical, but sometimes it's harder to find some resources more than others, I would say.
Yeah, I agree with a lot of that. I want to focus on, I think, two other areas that I think WashU does actually a really good job on, and continue to work in those areas. One gap that existed, I think, maybe more than a few years ago, was access to human tissue. To actually know what you're studying in the laboratory is relevant, to be able to touch the real disease that you want to have an impact on. And we now have an amazing number of biorepositories that contain clinical, genetic, blood data, as well as the actual tissue, cancer, tissue from patients with heart and vascular disease, neurologic disease, and many others. And I think that's a great resource we have that really doesn't exist at other institutions in such an accessible way as it does here. And I believe there's an ongoing opportunity and initiative today to try to do that at a much bigger scale, where we can create a cohort within our healthcare system that we'll have a very large biobank for, including genetic data for GWAS studies. And that's something that's being led up by MGI and Phil Payne and others here. And I think that'll be a great resource coming in the future. The second area that I think that is a big gap is we all have great ideas in the lab, right? I think it's pretty easy to identify a target that you think if you hit that target, you could have an impact on the disease you're studying. But how do you make the drug, right? How do you make the therapeutic? That's a lot harder, right? We all can do some initial screens, get some initial hits, but how do you develop that to something that actually is potent, is safe, and can go into a person? And that's a huge gap in the world of academia. From an institutional standpoint, there's a number of programs that are being launched here to help us bridge that gap. One of those is NPIC, the Needleman Program of Innovation and Commercialization that was launched earlier this year. And there's others that are coming down the pipeline. And I think that'll be a really important opportunity for each of us to not only do the first round of screens to get a chemical or biologic that may hit your target, but actually develop it to the point where we can put it into patients.
Just really quickly to add on to that, also, you can't put the same thing that you put into an animal model into a patient, and so there's a large and sort of unrecognized manufacturing kind of gap, and we do have some resources that are now being developed here to actually do that manufacturing in a way where you would be allowed to put it into a patient. So if you develop a therapeutic and it works great in tissues or animal models, you can't take that same thing and you can't manufacture it the same way. It has to be done in a much more rigorous, clean kind of setting before you can put that into a person. And most people don't think about that. They think, oh, I've got this new thing, particularly some of the biologics, so antibodies or cell-based sort of therapies. They have to be manufactured in a very different way, and it's very, very expensive actually to do it.
So just thinking about those regulatory, what are some of the hurdles that you've encountered and ways to get across to find that industry partner, or and when do you drop that onto the lap of industry? Where does the transition happen?
So I guess I can comment on that. So we ran two different types of trials. The first trial we did was using the existing drug, so that was the drug repurposing trial. The second one was the trial using a new drug developed by a biotech company, and so that was a new drug. And for both studies, we need to secure the orphan drug designation because I'm working on a rare disease. And to secure the orphan drug designation is quite important to get additional funding, both for as the academic investigator as well as the industry partner for the industry. And so for both cases, actually, ICTS helped me to do that. So I didn't know how to write up the orphan drug designation application. And so that was probably 8 years ago. I approached ICTS, and so ICTS referred me to CCS, and then CCS helped me to write up the orphan drug designation application. So that was incredibly helpful, and we have a very strong infrastructure. And the first IND was also written together with ICTS, and ICTS even provided some funding for that. So I think that's probably, so my point is to write up the orphan drug designation application and the investigation and new drug application. Both of these are required to start a clinical trial for any rare diseases in general. And you may feel, many investigators may feel that, oh, I don't know what to do. But, we have, you need to know that we have very strong infrastructure. And also, we have many investigators who have done it in the past. So I think if ICTS has a list of investigators who have done those in the past, that would be helpful, because then they can contact us. Then I am, or other people, I'm sure, are willing to help.
Great. No, I'll definitely make a call out to the Center for Clinical Studies. That is a great support. And I think a lot of us can help navigate and identify. Obviously, the folks up here are great resources, but there are bigger networks out there too.
So also then commenting on when to engage, industry partners, I think that this completely depends on you and what you can achieve, because you're going to know when you hit the roadblock, right? So for us, it's pretty early. We license our things to companies fairly early because we don't have the in-house medicinal chemistry. You know, we don't have the capabilities of doing PK or formulations or getting it actually into humans. We can get into mice really well, but that's where we stop, and I know that that's our boundary. But other people have the capabilities to take it further. So I think, you know, if you're committed to translating, you hit the, you know, the point of where you can't move it forward. Start engaging partners as soon as you can, because you don't want it to stall out. My advice.
Anyone want to comment on kind of the conflict of interest or that arises in doing a trial on something that you've really put your vested interest and expertise in?
So I'll start out and talk about that a little bit. Let me just define the conflict of interest so everyone knows what you're kind of talking about. So say you invent something, come up with a new therapy, a new diagnostic test, and you have a patent that involves you, right? Because you have some ownership over that. And you need to now test it in a patient population. You can't run the clinical trial, right? Because you have a vested interest in it succeeding. That's the essence of the conflict of interest. And so the way that's typically navigated is you identify a partner, right, that can run that clinical trial, who may be an expert in that area, content or disease expert. And that's why team science is so important. And I think the concept of team science, in my opinion, is a great way to navigate the conflict of interest.
I feel that, you know, that usually, you know, especially physician scientists, they may find something in their own lab, then they know the molecule or pathway very well. You as a person who knows the molecule and the pathway, then you also know the disease very well. Then you should be the PI for the project. But if you have the intellectual property rights of the molecule, then there's a conflict of interest. And then in many cases, university tells us that, you know, oh, you have a conflict of interest, so you should do a trial at a different medical center. Or if you set up multiple sites, you could be still involved in this study, but you cannot be a PI. You have to find the kind of a surrogate PI, and you can be a member. So I actually don't know how to get around with this issue, but hopefully for rare diseases or I think the university makes an exception. That's my hope, but it really depends.
I guess I'll just comment. I don't have any solutions to this at all, but it is a bit problematic because you can do a lot in the basic science kind of world and you know, develop things and everything. There's a lot of talent and stuff at CCS. There's a lot of talented clinical investigators and clinicians who need to be involved in sort of moving it forward. And so this current strategy of saying, okay, well, you have to be completely divested of any interest in this, means that either you're, you know, sort of taking somebody and telling them they need to do this without having any real investment in it, which sort of seems unfair and puts a bit of a problem, you know, it puts an valuing of the clinical research side of it to some extent. But we do need sort of things in place so that participants are well protected. The other thing that comes up is, yes, there's definitely an individual sort of conflict of interest, but if it's something that you developed at Washington University, then the university also has a conflict of interest. So managing all of that, you know, now if the whole university has an interest in developing something that maybe isn't, and, you know, for a small patient population that maybe is something that, you know, could be used to treat a large and important medical condition, figuring out how to move that forward, I think is a question that we still need to have a lot of discussion about.
I can give a little more background. I'm involved in 2 situations like this, where I was involved in developing the therapeutic that's being tested in people. So obviously I have a conflict of interest. My colleagues in the field I think they're pretty smart. I'm not a trialist. That's the other thing, is that as a basic and translational scientist, I'm not a clinical trialist. That is a completely different skill set. And it's very useful to have a trialist run the trial. You can certainly be involved in helping develop the clinical protocol as an advisor and being involved in the study as an advisor, but you can't be the responsible one. And at first I felt like I'd be left out a little bit, but actually don't in any way, and feel part of a team. And I actually have learned a lot from the trialists of how to run a clinical trial, how to think about it in a different way. And I think it's good to get other people's opinion because it gives you a better vision of how a therapy like what we're working on, if successful, could be implemented. And sometimes you come out with a different view than you came in with. And so I found it a valuable experience.
Kind of a general question about community and getting the patient voice in what you're doing. Is that a challenge, or are there ways that you've been able to get the community involved?
So my team has been closely working with patient organizations because I'm working on a rare disease. And so there are 3 patient organizations in the United States and also several patient organizations in Europe. And so we have been closely working with them for 2 reasons. The first reason is to know what our patients want through the clinical trial. And initially, so then what we did was we worked with the patient organizations and tried to identify the outcome measures patients want. And so we sent out emails to several hundred patients and to see what's their most bothersome symptom they feel. And then they said, low vision. So then we really focused on, we tried to create clinical trial protocol around their low vision. And at the last moment, due to the feedback from FDA, we actually have to change our clinical trial protocol. But it was quite important to do that process. So it's very important to work with patient organizations to establish a protocol together. Also, we are currently conducting a clinical trial with a new drug. So what we did was we created a patient board. So from time to time, we show the data to patients, then ask their opinion. Also, patients themselves are working together to actually appeal FDA about their ongoing clinical trial. So I think, especially for rare diseases, it's very important to work with patient organizations or patients to get their feedback. And sometimes they help us maybe extend our clinical trial or expand our clinical trial or even for approval of the medication. So I think it's critical to work with patient organizations and get their feedback.
It's also really, really helpful to have a community advisory board, and I know ICTS has one. As part of the AIDS clinical trials group, we've had one for, 30 years, and they're very opinionated, and they're lovely. And it's actually, what we've found is really, really important is to have the patients review the protocol. And I think a lot of people feel like it's too scientific, it's too complicated and stuff. You can train people to read a protocol, you can give summaries and everything, but the most important thing about having patients or potential participants read a protocol is they will tell you, not doing that. They will also tell you, like, this isn't gonna work, the timing of this isn't gonna work. I mean, that's the other reason to have people who do protocols also involved, because a lot of times the people who want to develop the drug and make it perfect are involved in writing the protocol, and they want the Goldilocks patient, and they don't exist, right? So you have to think about, like, how do I make this protocol accessible to the most number of people so that it's actually relevant when you actually publish the data. So yeah, and community is great with that.
All right, I'm going to ask one more question, and then I'll definitely want to open it up if people have questions here. Someone had mentioned just financing, and how do you get the resources to do this work? And just wondering if you could comment on that.
So I can also comment on that. So initially, our study was supported by donations. So initially, my study, both basic science and also even clinical trial study, were supported by donations from patients. And then using the data obtained from the study supported by donations, we ended up getting the grants from NIH, and also we also got some funding from ICTS actually. So I think there is no straight answer to finance our studies, but by closely working with patients and to gain trust from patients, I think we have a chance to get support from patients through the form of donations. Then by using donations as a stepping stone, we can get larger funds from either NIH or industry. That's what we have been doing.
Yeah, I mean, I think the NIH does have quite a bit of funding to enable at least the early stages. Once you go into the clinical trials, it's a whole nother world. But at least getting into the clinical trials, there are a lot of grant mechanisms at various steps, including partnerships, like these small business awards, that it can be partnerships between industry and academia. And so those have been really instrumental. But I also want to echo kind of a point that was made, is that I feel at least that I've had to really go out there and talk to people, network, and fundraise. And maybe it's because, if you're really passionate about this, maybe you are the best person to be talking to people, getting donors, engaging with patients in certain disease areas. But I think you have to be ready to be a fundraiser yourself. And there are some resources here, but you will probably be the most effective fundraiser in the other partners that you're working with. But there are grants. ICTS also, thank you very much, has funded us as well. And so there's opportunities, but to really get to the big level, to get companies interested, to get those clinical trials done, I think you have to put yourself out there some.
I'll let Kory take the last one, but yeah, I think it's a patchwork. And that's probably part of the problem, right? Because a lot of times it's a patchwork of a lot of small amounts, and this kind of stuff is really expensive. So you have to find different ways to move it forward, and yeah, I think it's always a combination.
So funding is an issue. It's a major issue, and I think we all need to recognize that. So I think identifying targets, getting your first kind of compound can be done through NIH studies, or NIH grants can get started through ITS as well, and other sort of foundational grants. But to really launch and to make a therapeutic that's potent, that's safe, that's tolerable, that can be manufactured in the right setting, to go into the patient requires money that's outside what the NIH can do. And I think each individual person who looking at their program needs to decide whether that's something that needs to be done in their own sort of small business, in partnership with biotech or pharma, or could it be done within the university to some extent. And there are university initiatives like NPIC that are trying to bridge that gap to get you to a point where whatever you're making is of enough value and you have enough proof of concept data that you can really put in a person. I do want to highlight, see Pam over there, so I want to highlight that this problem has largely, in some ways, been solved in the radio tracer space, in terms of making molecular tracers that we can put into patients. Our Department of Radiology in Mallinckrodt has done a great job in having funding available and resources available to design different tracers, to make them in the GMP environment, even in preclinical studies, and to do first in-patient studies, both single center and multi-center. So I think, given their example, this can be done, and it can be done in the university.
So I'd love to have, if Pam's willing, how did you make it happen, and is that something we could translate to other areas? So I'd love to open it to people around here in the room who have questions, but Pam. Thank you.
Sorry to put you on the spot.
No, that's quite all right. And I think it is a little easier for radio tracers because we have several FDA mechanisms. Sorry, I'll stand up. FDA mechanisms such as an EIND. So in terms of tox, the tox is not quite as rigorous. We're injecting very small amounts, less than 100 micrograms. So, but yes, it is possible. We do have people who can navigate the FDA environment and answer questions from the FDA reviewers. So happy to help, especially, for any imaging agents. We do have a lot of expertise in that area.
We'll bring the microphone to you. And if there's any other questions online, put them in the chat and we'll.
Yeah, so you guys talked a lot about the first gap, the gap from, we'll say, bench work to the clinical trial work. My research has mostly been from the second gap, so from the clinical trial to then implementation. And so a lot of talk here has been about, not all, but a lot of talk has been about how you fund and support the translation from bench to clinical trial. And I just wanted to point out or remind the audience that the choices you make there have implications for the second gap. So some of the choices you make there, if you partner with industry or a pharmaceutical, not always, but sometimes you're going to make the cost so high that the second gap is non-transversable. So keep the policymaker or the payer in mind as you're making those first decisions.
I wanted to first thank the discussants. That was a really rich and useful discussion, and ask a question about drug repurposing. We got a very dramatic demonstration early in COVID that can often be the fastest route towards getting treatments out into use. In particular, the United Kingdom recovery trial within months of COVID had proven and disproven several treatments and was estimated to have saved 1 million or more lives in the effort. So my sense is that there's a lot less appetite for repurposing drugs moving into the clinical trial arena in the US, probably around skepticism about intellectual property and making money, if I can be honest. I wonder, and I know some of you have thought a lot about this, I wonder what your thoughts are about that. Is there a pathway for drug repurposing in the US?
I can make a few comments about that. I think it's a great point. And a lot of times we think of drug repurposing around, small molecules. But I think there's been a lot of success actually in repurposing in biologics and antibodies. There's a lot of theft from all the rheumatology antibodies and other fields that have been dramatically successful. I think one great example that I think speaks to the issue of patent protection and whether it's actually feasible is Regeneron many years ago made an IL-1 trap called rilonacept. And they tried every single indication they could think of on Earth and failed everywhere. And then they looked to try to sell it. And so they sold it to this tiny company called Kiniksa, who tried the drug in a very rare disease. I bet people that are in practice in the room have maybe seen a few cases. Chronic recurrent pericarditis, basically chronic pericarditis that recurs, that's it's not responsive to any of our current medications. And in a trial of 62 total patients randomized, it was a dramatic success and they got FDA approval. And I think we're seeing more and more examples like that in the biologic space. And that's a little bit easier for us in academia because we can think of it as being on mechanism, because you know exactly what that mechanism does, and it's not the mystery of a small molecule that may be hitting something else due to a lack of selectivity. And so I think we need to keep this in mind. I think it's a pathway in academia that can be really successful because that therapeutic has already been vetted. It's already been shown to be safe. It's already been shown that it can be put into patients. It can be manufactured in a safe environment. So I think it's a great point that you bring up.
So I agree. So I think there is a pathway for repurposing drug to get the approval for any disorders, including rare diseases, and we are working on the rare disease. And we ran 2 trials using, we did we did run 2 trials using existing drug, which was drug repurposing trial, and we are currently running the industry-sponsored trial. The difference between these two is clearly we get less money we got less money for the drug repurposing trial. And so we wanted to expand, but it was very difficult. And so I hope for the drug repurposing trial, I hope we actually discussed if we can so it costs so much money for the disease like we are working on, because it's a syndrome, so we have to look at different domains, and we invite patients to our trial, our trial site from different states, even from different countries. So it costs so much money. But if we can assess the efficacy of medication remotely, as we discussed, if we can come up with a good way to assess the efficacy remotely using iPhone or tracking medical records, that's going to significantly reduce the cost, and that's going to increase our chance to get the approval for the repurposing drug.
I think we need to do a lot more also with repurposing, because there are lots of problems, especially an older drug, right? There's no financial incentive for a company to partner with you. But I think it's a huge potential opportunity to move things forward, and to some extent, it's also what doctors do all the time. We use a lot of drugs that are not for the indication that and it's really, really important in academia to actually prove that those are doing what we think they're doing.
Great. Sam?
Yeah, I'd also like to congratulate the panel on a great discussion. I wanted to make one comment and a pitch as well. And this is an expansion of what Kory Lavine had mentioned. I think a huge gap here is a translation of relevance of what you find in basic science of humans before you go into clinical trials or doing any kind of clinical manipulation. And to do that, we have exceptional resources in our clinical translational research unit to do small studies to look at mechanistic effects of pathways you might identify in rodents, and not just based on gene expression data or repositories, but actually testing it in humans by using either repurposed drugs or other drugs to see if those mechanisms are actually relevant. And our Institute for Clinical Translational Science, I think, has been one of the best advocates of a research unit in the country. Many other research units have not been supported like ours. We also have core services like our Nutrition Obesity Research Center, where you can collaborate with expertise to help you design those trials. And we have two studies right now in junior investigators that are testing the relevance of what they found in rodents in people in small numbers of subjects, looking at mechanisms, not clinical outcomes, but metabolic mechanisms that then you can translate to clinical outcomes. And this would then imply we should support even further the Clinical Translational Research Unit by Dr. Powderly, which I'm sure he's planning to do in the next cycle.
So I also echo the excellence of the panel. Thank you. I just want to make a comment about repurposing drugs, that there's a lot of interest outside the U.S. in repurposing drugs, and so often if you're having trouble getting funding, think about finding international partners, other countries where you may be able to do your clinical trial. Find a few centers in the US so the FDA is convinced it works in Americans too. But especially if you don't have a rare disease and you're going to need substantial numbers of patients, that's a really effective way to do a drug repurposing trial.
All right, I want to ask one final question, and I always ask my patients this. So if I had a magic wand that could help get you past one gap, what would it be? What's the biggest challenge that you see in your own work?
I think information. I mean, I think the more examples we have of people who have gone through that gap and actually moved things into patients, and having that actually available to people so that when they hit the wall, they don't think, oh, no one's ever done this before. I think that would be, I mean, money always helps too.
Yeah.
I think time. I wish I had more time. That would be really helpful. But I think also, in addition to the information, the network, like just, which you have also already done like a good job, but just continuing to expand that network. And with all of this like GWAS stuff coming around, like building the network and capabilities with that, like experts that can enable use of AI and actual analysis of these tools. So not just having the resources there, but having the network with the expertise that can just make it all happen would be amazing.
Mine's a bit more boring than that. I think it's resources around hit to lead optimization. So I think it's fun to find a hit, to get a early molecule or antibody that hits your target. It's really boring and really, really hard to make it better and safer and more potent. And frankly, in academics, we don't have the expertise to do that. And so building that expertise within the university, I think, would be a great thing.
So in my case, actually, I always have some, regrets when we choose the clinical trial participants. And so to identify the proper or best patients in which we can see the efficacy of the medication very efficiently. I think that to identify these patients is quite challenging. And we need to know the disease very well, and we need to know, should we choose the patients who have mild manifestations? Should we choose the patients with severe manifestations? Should we choose the patients at early stage of the disease or late stage of the disease? We are always not so sure. So I think if we have a good way to identify the best patients to see the efficacy of new treatment, I always feel that's quite a challenge.
Bill?
So I wanted to thank all of you. Chris actually asked the question that I was going to ask, what the institution can do for you, because I do think it's really critically important for a symposium such as this. But I want to take up one comment that Kory made that I think is really, really important, particularly for people who are at the very early stage. And that is that involving early in the process, if you think you're going to ultimately go into clinical trials, involving the clinical trialists, because the way they think about how you will get your study done is critically important. And I want to take up also the comment that was made earlier, is that we are also we have probably the best implementation science group in the country, and we also look at the question of thinking about not only when you do the clinical trial and you show it works, and how you can implement that in a wider population, and involving them in the clinical trial process early. So we actually have a small grant process, but if you have an existing clinical trial or developing it, we will fund the implementation science person to come work with you to think about this. And it's really, really important. I'll give you one recent example that Lavine Gang, who is the director of our Implementation Science Center, told me about. And it wasn't an institution here, he was at another institution being asked to give advice about a really promising drug to treat HIV infection. Really, really promising. And the place was going on to start proposing clinical trials. And he asked a very simple question: how do you need to deliver this drug? And they said, well, it'll have to be driven intravenously. And he said, this is a pointless exercise to go into a clinical trial when you already have patients where the standard of care is one pill once a day that delivers 3 medications in that one pill, and that where the field is moving to monthly and potentially quarterly injections. So a daily intravenous therapy ain't going to go anywhere, even if it is the most wonderful thing in the world. And so there is this whole concept of bringing it, and I think Kory expressed it really well, that we have incredible expertise at a place like this. We have people who know the disease well. They may not be clinical trialists, but they actually treat patients all the time. We have people who do clinical trials, and we have investigators who are really want to advance the field. And one of the mechanisms is to get them together and think about how do you do this? How do you close this gap? I think you've identified some of the structural issues, but there's an intellectual issue as well. Part of translational medicine, I say to everybody, is actually getting everybody to speak the same language.
Great. Well, with that, thank you very much to the panelists, and we'll move to the next topic.